Hi, today we are covering mitosis, and that is from the Greek word mitos, which means thread, and that's referring to the spindle fibers that are pulling our chromosomes apart during mitosis. I'm not sure how to get the video synced up. It's moving either too slowly or too quickly. Okay, there we go. From the Greek word mitos or thread, again referring to the spindle fibers. So the process of cell division and replication. So this is our normal cell replication, healthy cell replication. And it occurs in all somatic or body cells. So when we're growing new bones, new skin, replacing things that are damaged or getting old. It's actually kind of a short process of the life cycle of a cell. So it's just the end set of stages at the end of the cell's life cycle. Most of the time, the cell's just happy living in interphase. So after mitosis, interphase happens and the cell is smaller after it divides. So we have growth at first in interphase, and then you'll have synthesis. So that's the S stage. So G1 is growth. S stage is synthesis, where DNA is replicated. So it's still all strung out as chromatin, but then it does duplicate. So you have two copies of everything. And then growth and production of enzymes and production, like proteins, that will be necessary for the process of mitosis. So that little piece of the pie in the cell life cycle is mitosis. So mitosis has four main stages that we're going to focus on. So there are some people that add in some like pro-metaphase and things like that. We're just going to go with four stages, and the letters are PMAT, or PMAT, prophase, metaphase, anaphase, and telophase. Okay, so you can just memorize it as PMAT, to keep it in order. So prophase is the first phase, and you can see that the nucleolus disappears. The nuclear envelope is actually disappearing as well. And we'll pause there like a little bit so we can reflect on prophase a little bit better. Okay, so you're gonna have creation of these mitotic spindles. So up here you're going to see mitotic spindles. So the chromatin, which is just like yarn-like strands of DNA spread out, we want to realize that it's no longer spread out. It's condensing into these tightly wound chromosomes, so that it's like packing your laundry into a nicely folded suitcase so that you don't have things strung out all over at the airport. So with cell division, you're not going to have your loose yarn ball of DNA. You're going to condense it down into these nice, tightly wound, very nicely packaged chromosomes. And that only happens during mitosis. So prophase, you're going to start seeing chromosomes, and you're going to dissolve this nuclear envelope and form the spindle fibers. Those are the key characteristics that define prophase. So we'll go on from there. Okay, so prophase is the P, and then we have metaphase. So metaphase is kind of an imaginary line. So the metaphyseal plate or metaphase plate, it's like our equator from the Earth. It's like there's not like a distinct landmark there. It's just a chemical plane where all the chromosomes are lined up. And the reason that they do that is so that you can pull one to this side and one to this side. So we duplicated our DNA, right? So we have now two complete versions of our genetics. So we just need to pull one copy to this side and one copy to this side. So you'll see the spindle fibers and then everything lined up in the middle. That's metaphase. Okay, so just another version where you can see the chromosomes all lined up on that metaphyseal plate. Then we have anaphase, And anaphase, I really do like to visualize just a little girl named Anna just pulling the chromosomes apart. So it's in the process of pulling apart, so they're not in the center like in metaphase, and they're not completely at the poles like they would be in telophase. So that's our next phase. So anaphase, they're in the act of being pulled by those spindle fibers towards the centrioles at the poles. So we want to make sure that we aren't confused, like whether they're in the center or whether they're completely condensed at the pole. And I'll show you one more trick as to why you can tell the difference between anaphase and telophase. We'll click on telophase. Okay. So telophase, you are actually going to see them condensed at the poles, and this picture doesn't really show it at the poles very much. But one very distinct thing is that you're going to see those clear, distinct boundaries start popping back into place. So that nuclear envelope surrounding the genetic material, that will start coming back into place. So now we have one cell. It's a really long cell, but it has two complete nuclei inside of it. So that is telophase. So you pulled your chromosomes to the poles as far as you could get them, and then started re-enveloping or recovering or re-enveloping your genetic material. So that's how that worked. Okay, telophase. I apparently left this slide really long. Okay, cytokinesis is the actual splitting of the cell. So you'll see a cleavage furrow form. It kind of puckers in. And then it's just contractile proteins pulling that cell membrane together, and so the two cell membranes will actually end up binding here and creating a complete cell membrane. So cytokinesis is the final stage. Traditionally in lab, we look at an onion root tip. So we have all the phases here, if you can recognize them. So we start with prophase, and you can see that the nuclei is kind of dissolving. So this is what it traditionally looks like in interphase, and you can see a clear nucleolus inside, and you can see one clear nuclei. So we'll go back a little bit. I have to drag it. Okay, so you have interphase, where the cell's just normally living its life, right? So this one's really, really clear and distinct. You have your nucleus and you have your nucleolus. So it's like an egg yolk inside the nucleus. And so when everything's normal, that's what the cell living out its life looks like. But then when we're going through mitosis, we remember prophase, we started condensing down the chromosomes into, well, your genetic chromatin into chromosomes, and they're not really organized. So you can see it, and you can kind of see the nuclear envelope dissolving, but nothing else has changed. Like they're not really in any structured format or anything like that. But then metaphase, they line up on that metaphyseal plate. Okay, so you can distinctly tell that. There's another one over here where I would still call that metaphase. Then when they're pulling apart, you have anaphase. They're not at the poles, so you can tell the difference here between anaphase. I would actually call that telophase, but when it gets really, really, really condensed, and the reason that this is telophase is because you can actually see those membranes of separation forming. So telophase, you have your chromosomes really, really pulled tightly to the poles, and then you have the membranes reforming around it. So you have a nuclear envelope forming, and then there will be the cleavage furrow, and the whole cell will actually divide. So those are really good examples. This is the lab that we would have done had we been in the lab, is looking at onion root tips. Okay, and just one more really, really good example. Okay. So here's one more really, really classic example. So here's it just drawn digitally so that you can see prophase is the chromosomes condensing down and the nuclear envelope dissolving. And so this is interphase, where everything's strung out, and you can see it condensed down. So normal nuclei and interphase, what it looks like as it's moving into prophase. Then we line up on the metaphase plate in the center of the cell, and then when they start pulling apart, then we have anaphase. And as we completely pull to the poles and start making those membranes to divide inside, so you'll see the reformation of the nuclear envelope, and you'll see a boundary where these will pull in, and you'll have two cell membranes inside the cell to completely divide. That is mitosis. So this is normal, healthy cellular reproduction. And it's kind of crazy. I'm using Powtoons and Screencast-O-Matic trying to make these videos. So sorry for any of the glitches, but I hope that if you have any questions, you will email or call or leave comments below, and I will try to respond. Okay, we'll talk to you guys soon. Bye.